I'll probably ask a little bit more specific in the final because we haven't covered it. But as an example, I would probably look at the chapter, because what I'm going to do is I'm going to go through the chapters at the end and say, well, what's really important in this chapter, and I think I'm going to have more, maybe 50% essays and 50% multiple choice. People do better on essays and it shows more what they know. And so I would say, in this case, I could say,
50% essays, what you said?
well, so instead of like short answers, how would I, definitions, how do you ask for 5 to 10 definitions if you want to know what somebody knows? So it's more about major concepts, and I would say, in this case, I could ask something like, tell about the 3 lines of defense in the immune system or something, and just write down the things you know about each, or just so you have an idea. It should be more of a general approach to each chapter. Or if it's, say, about, say, chemical and physical methods to eradicate or to limit microbial growth, I could say, explain a few of each, or have a few examples of each, and how do they what's their modes of what are their modes of action, things like that. So it's more, just want to make sure you know that it's more general. And I'll give you, once I'm through it, should be by Thursday, I'll write out, what the major concepts of each chapter are. And you don't have to go in and learn a definition for, I don't know, some endotoxin that some bacteria produces. Just gonna forget that. So, so remember, for the first line was all the fluids and barriers that we have in the areas where we are exposed to microbes the most, and then the second was the innate but not acquired. So it's not acquired. So it's the defense, the second line of defense or immunity that we're born with. And there's certain parts to that, like the lymph system, the blood system, that carries a lot of proteins that help in immune response. The reticuloendothelial system, that is those are areas where a lot of the immuno cells, the cells that the immune system has, the white blood cells, where they're located, and then the extracellular areas where a lot of those are also located to take care of microbes. And so mainly what happens in the second line of defense is inflammation is a possibility. You've all had it, where you get inflamed or you have inflammation, and say I think there's an example of inflammation of a finger. And then phagocytosis, meaning somehow those whatever bacteria gets into your body, whatever microorganism is being taken up by specific white blood cells. And then there's 2 others, and that's interferon and complement. And those are basically, complement is just blood proteins that are in your blood. There's more than 20, and they kind of recognize there's different things they recognize, and we go into that, but they basically help identify foreign particles, and then actually work together with the specific immune system. So that's complement. And interferon is basically, again, it's just a more local response, where this product of one cell, so one cell gets in contact, say, with a virus, and then produces this, and signals by interferons to other cells around it that it's being exposed to something, and then the other cells can prepare for that virus. But those are not specific, so they usually recognize patterns. And remember, a lot of the microbes have these pathogen-associated molecular patterns. So all you can think of is like if you remember the virus, a lot of the virus surfaces or proteins that you find on the outside, or the bacterial surface molecules. Those are the ones that are recognized, but they're patterns, so they're not specific. All these responses are not specific. Later today we look at some really specific, where a cell is basically, or one cell in your immune system can actually recognize a very specific cell, and it's just that one cell. And then that one cell starts making a lot of cells that are exactly the same, so it produces all these clones, and then these clones attack a very specific antigen is what that's called, but we'll get to that later. Just so you know the differences, or you have an idea of what the differences are between the innate and the acquired immune response. So if you go to inflammation, you have the usual, and that would be another one where I'd say, how do you personally experience it? And you experience it probably just like that. You have first, if you have an injury, say you get caught up on a thorn, where initially you actually initially have like not a vasodilation, it says here, but you actually have a constriction in the very first few seconds. But then usually it dilates, which helps bring in more blood and bring in all these other cells that can help with attacking microbes that are getting in through the wound. And then because you have increased blood flow, it gets warm in that area, so you have first a very short period of your vessels constricting. That's very quick, a few seconds, and then they actually open up, and then you have blood flow into that area, which makes it, number one, it makes it warm, and then it swells because you have a lot of materials migrating into that wound area, and then which you would call an edema. And then there's the white blood cells. You have a mix of white blood cells, microbes, debris, fluid, and all that, and that forms the pus that you usually can find, and it prevents the spread of the microbes. And then the pain that you have, it comes from just the stimulation of your nerve endings. So that would be an inflammatory response. That's one of the responses that allow the white blood cells to move to the site where microbes are expected, because now you have an opening, just like the person that used the dirty needle for injections, or other scenarios where you have an opening to the outside, and your protective skin layer, the barrier, the physical barrier of skin is not intact. So it shows here again an inflammation. We just went through this, but I don't know if you remember the mast cells, and you can, if you want, leave, depending on how you follow the lecture, but you can leave the cell differentiation, the white blood cell page open if you want. I usually look back to which cells we're talking about. The mast cells were cells that we said are located in the tissue and are the first responders, but they're usually, so they're already there. They haven't migrated in, but you have the injury, and then you have a short period of vasoconstriction, meaning your vessels close up, and then they open, especially because you immediately, or you start forming this clot, and so they can open up. You can have a lot of different white blood cells move in, like these neutrophils, and then that, along with the bacteria, produces the pus we talked about, and then the final, and kind of an edema, which means you have a lot of liquid in this area, or a swelling. Fluid is moving in. That's what they usually define as an edema. And then by the end of it, you have resolution. Usually it forms a scar, so where this clot is, it becomes a scab, and then you have a little scar there. Eventually goes away if it's a small injury. And then you have finally also lymphocytes moved in. So usually you have some first responders, and these mast cells usually just release chemicals that signal to other cells to come to this site, and so it opens up. The blood brings these other white blood cells, and then they can take care of the microbes that came in, and then you also have blood clotting. So that's a typical inflammation. And you can think of how it works in your own body when you have a cut or something, and then you kind of see the same pattern, except you don't know what really happened, how many microbes came in. So you can kind of think of those now, being exposed to them here more often, and think of how your body responds to those without you even knowing. And I don't want to go too much into this. It's just basically what I just showed you. You have an injury, which is some trauma, and you have vasoactive action. So you have a short period of constriction, then a dilation, dilating the blood vessels, and then you have a lot of chemotactic action. So that means you have some cells like I showed you the mast cells releasing mediators over here, and then you have major phagocytes that come in, macrophages. We talked about those, the biggest cells. Those are the ones that can actually engulf these microbes. And you have a lot of cells migrating through vasodilation. And then there's some examples. You have chemical mediators. So those histamine you probably heard before. There's a few, serotonin. They have other important functions in your body, but they also have these effects on the constriction, or basically on the tension on your blood vessels. And there's a lot of flexibility in how much blood flow can go into specific areas. And then a lot of these a lot of those substances that have chemotactic effects come from other cells. So that means chemotactic means they're basically there's a signaling molecule that allows other cells to move into a specific area. Chemotaxis would be a specific substance allowing or signaling the move to a specific area, which is the area where you expect the most microbes to be. So leukocytes have what's important then. If you think of what we just looked at, one thing they have to be able to is to migrate out of the blood vessels. So if you have a blood vessel going to a certain area and it opens up, and then you expect it to go to the site of injury, they actually are able to leave the blood vessel. So they come, and because of the cytokines, because of these different chemicals that are in the area, they're able to leave the blood vessel and move into the tissue. So it's a unique characteristic of leukocytes called diapedesis. And then chemotaxis, I just said, so in response to specific chemicals that are usually given off by the first responders. It's almost like you have a person with a cell phone seeing an accident, so they're really just there, and they call the ambulance, which are then the bigger white blood cells like the macrophages, and they come to the site, and they're able to take routes that nobody else is allowed because they're the ambulance. So they can get out of, they can drive out of, they can take off of the freeway through the grass and this is the freeway, that's the grass. No, I mean, you get the idea. It's just, I know you're gonna think of a crime scene next, or a accident scene next time I ask about it. That's good. Yes, don't write that.
Right.
They're gonna ask, so how was your first class? Here's an example of an exam. So the ambulance goes to the grass. I wasn't able to ride there. Another pretty common reaction of your body, and I have tried this so many times. Does anybody try to, you get a little bit sick, and then what I do is, what I've done before, I got so much grief for it that I don't do it anymore, but I used to bundle up with a sleeping bag and then stuff over it, and I get so hot, because it worked once for me, I don't know why. I was really cold, so I put it all up, and then I sweat so hard, and then I came out of it pretty nasty, but then you take a shower and you're like, oh, it's 12 hours and I'm done. This everybody else is laying down for 3 days. So now I do it with the smallest things. I just go in, and then I get in such a bad condition that I don't know what's worse. And people say, I don't know, it really worked the first time. It was the flu or something, and I thought, man, I thought it makes sense. I didn't know much about it, so I thought, well, I'm just gonna, I'm basically, my body temperature is so high that nobody can stand it but me. So everything else is going. Everything else in my body is dying. But then, I don't know, it lately hasn't first of all, hasn't worked, and if you're not single, if a family watches you, they're like, nobody's tried that before? No?
I have heard if you're feeling a fever come on, not to take Advil or Tylenol, because your body is trying to heat up enough to kill off the toxins that can't live it.
Well, there is some justification for this. I was just wondering if there's numbers behind that too, in terms of students trying it out or people trying it out, because I never knew the background. I just remembered, for some reason, I might have been cold that first time and just did it, and then I came out and I thought, man, that's it, I got the solution. And then a few other times, the next 5 times it didn't work. But there's some points towards why I'm probably right, and it just didn't work the last few days. In a little bit. So what they call that is pyrogens, and the hypothalamus, a structure in your brain is usually regulating your temperature, and what it does is it signals to the muscles to produce heat, and so you have basically muscles having smaller contractions and producing heat, and also vasoconstriction. So your vessels close up, and you have more heat in the core and not so much in the small capillaries in the outside in the suburbs of your body. So there's 2 different ones, just the exo and endo. But basically, if they come from if this whole process of heating up your body, of fever, are products, if that process is initiated by products of an infectious agent, then it's exogenous, makes sense. And endo is if that is initiated by your own white blood cells. So your white blood cells realize something's going on. They signal their cell phone calls to all kinds of places and stations. And there's also a call to the brain to say, you have to heat up the body. And so the benefits of that fever, which I probably have a poster at home now on my wall, is you inhibit the multiplication of temperature-sensitive microorganisms, which maybe that's the problem. I just didn't have the right microorganisms because they weren't temperature-sensitive. It also changes the availability of nutrition for bacteria, so you imagine there's less growth, and it increases the metabolism in general, because higher temperature means things are usually, until you come too high, things are speeding up, so your immune reaction, your other physiological processes are sped up, and so you could you imagine that helps fighting microorganisms. But it's usually not you know, it's just in a certain range. If it gets too high, it gets dangerous. If you stay up at a higher temperature too long, it's dangerous. But it definitely has benefits. And maybe the fever just has to run its course, and you don't have to help it. That's the conclusion. So phagocytosis is one of the things. So we went through inflammation, fever. Phagocytosis is another process that helps in your defense. And you know, macrophages are there. Neutrophils are engulfing and destroying microbes. So what usually happens is the general activity is they move around and survey your body, and there's the lymph system, there's the blood system, so there's cells moving around, and they look for microbes, they look for these pathogen-associated molecule molecule patterns. So they look for you know, proteins that are displayed on organisms that you don't want in your body, and so they find those, they ingest and eliminate the material, and some, which is a really interesting and important part, and we'll get to that later, some actually take that, a specific structure and what we call an antigen, and we'll get to that a lot more later, and basically extract it and then later on present it. So basically, the process, some cells are not only recognizing this is a wrong organism in the body, but they also store some of that material and say, if you want to help me, basically to other white blood cells, I got some. This is what it looks like. They basically keep a piece to show, and then these more specific cells, like the T and the B cells, which we talk about later, are then gonna see those pieces and produce their own specific reaction. So this helps. This last part helps to actually produce a specific immunity, and an immunity that is able to respond to a second, or third, or fourth, assault by a specific microorganism. And so these phagocytose capable white blood cells, and you can go back to your page, are neutrophils. We talked about those. Most of the cells, the white blood cells we have, their general purpose, and they're one of the ones that you find the most. They act early in damaged tissue, etc. And then I already said that eosinophils are the ones that are more geared towards eukaryotic cells, so they're more attracted to sites of parasitic infections, protozoans, and then the macrophages, which are the biggest one, and they're really important. So always remember, and they've got they're the ones that have the phage in it. So they're derived from monocytes, and you can see that on the page 2 if you go back to your white blood cells, and they help in the process of activating for B and T lymphocytes. So those are the specific T cell, the specific white blood cells. And just so you remember, you have a pro-monocyte. So this is how the macrophages come to existence, and you can see that again. If you go back to your white blood cell page, you have a stem cell in the bone marrow, and the pro-monocyte is the next step, and then these go through the blood and develop, going through the blood into macrophages and dendritic cells in the tissue. So it's just, you imagine in the bone marrow is where it all starts with your blood cells, red and white, and then some of them develop in the thymus, some of them develop in the blood, and some of them actually stay in the bone marrow to actually differentiate to what they need to be. But these are traveling these specific macrophages macrophages are the ones that are traveling in the blood and then actually in the tissue become these cells that can eat up, or phagocyte. What do you call what's the verb for phagocyte? They can ingest microbes. I'll keep it at that. So those are the different. So those are the few cells. So those are some of the cells that can actually ingest and destroy those microorganisms. And there's a few more. I mean, T and B cells do that too, but because they're part of the specific immunity, we're not talking about them now. Just in a little bit. So we talked about, or I already introduced some of those receptors, and I don't know how to do that best, but just keep in mind that you've learned of microbes and the different even in viral cases you learned about how they have specific proteins, and those proteins are different from ours, and so we recognize them as foreign because they have these pathogen-associated molecule patterns. But that's very general. And for those one of those receptors that look for those patterns is the toll-like receptor. But I don't really think that's too important. The most important thing is that if you look at a macrophage, it has a receptor which just happens to be a toll-like receptor that can then recognize this foreign molecule, and then what that does is it number one is probably going to try to destroy that any kind of foreign or it's going to try to basically ingest the material that's foreign. So this could be the molecule, this could be part of a receptor that's attached to a microbe. And so it's going to try that, but it's also going to, like I said before, it's also going to send signals out. And some of those signals are called cytokines. So maybe cytokines are the only ones that you should remember, but they're basically chemicals that this macrophage can produce and send out, and that's going to attract other cells to come to this area where where the macrophage suspects there's more. And how do they get there? Through the process of chemotaxis. So this is part of chemotaxis. They get the signal that something's going on in this area. So all these cells, I don't think you have to differentiate too much about that. All these cells are able to communicate. So if you have any kind of, if you think about, you get an injury somewhere on your body, there's some mast cells right next to it, there's some other cells right next to it, and they always call for help. And they do that by...
Bless you. Ooh.
How many brothers? You need to go home, lay right now.
Doctor's orders.
Doc, in the book where it says the bacteria will localize right around the point of, let's say, a puncture.
Yeah.
Can the white blood cells fight them right there?
Yeah. So that's where they move in, and that's where you have the production of basically pus or edema because you have a lot of cells moving into that area and then keeping the organism in place. And then they can either, not always they can eliminate them right there, but basically they're not going to let the organism move on. They're probably trying to keep it in this place. And sometimes you have swellings and they stay localized. And sometimes the answer is too slow, and then it becomes widespread. And then if it's systemic and you have it in other places, it's more of a problem. I remember we had localized inflammation or infections and broad problems. So in general, they do it right there. I heard I didn't hear that one, I read that. There was an interesting example, and It's not that, I mean, it's a little bit related to your question, but it was a viral disease, but it's in your book, and it's about rabies. I don't know if ever anybody read about it, but there was it's always really tough to read those cases because you think our medical, our professionals are so good that they can, once something is identified, they can do something. But rabies, which is a viral disease, is apparently like close to 100%. So creating or what do you call it, non-curable. Anyway, there was I guess in some cases, and I'm saying that because what it relates to is keeping it localized. Where something comes in is always important. So once it starts spreading, it's much harder to find it because we talked about how do you get to the cells that are infected with the virus. So in this case, there's a 9-year-old boy, a Canadian boy, who had a bat in his room that just came in. The dad kicked it out, and he had some little wound, and they didn't think of anything bad about it. And then 4 weeks later, he started having these crazy, I don't know what the symptoms are, but he had the shakes and he was dizzy and all kinds of things. So that was like weeks after he got bit by this bat. And then there was another kid who was 15 in South Africa, I think, and he got attacked by bats, which they must have been infected because their behavior was off. I mean, usually you don't get attacked by bats. Anyway, both kids had rabies, and the 9-year-old was so far into it, what happens is the virus travels from your skin and needs a long time, but it eventually makes it into your brain, and then in your brain cells, it's almost impossible to cure. So what they did with the 15-year-old, they knew exactly what happened because he went to the hospital right after the fight. He could have probably gone home and nothing would have happened for a few days or weeks, but they put him into an induced coma and then gave him antiviral treatment, and he survived, which is the first one, I think, out of they said a lot of people, and the first South African, and the 9-year-old couldn't be saved because it was so late and because it was, so if you have this response, and this is, like I said, it's a little bit different because you're talking about microbes that you see in the environment and that enter your body, but what the body is trying to do, obviously, is keep it localized, and then you can deal on a local level with it rather than having to find a cure that in a bigger area. The bigger the area that's infected, the more cells infected, the harder it's going to be. So I talked about this quite a bit. The chemotaxis, moving to it. So how do phagocytes recognize and kill the invader? And mainly it's chemotaxis. So they're sent to they know where to go because there's certain chemicals that are released by either microbes or also by mostly by white blood cells, and they recognize those pathogen-associated molecule patterns. And then usually what they produce is like something like they have a phagosome, so they have a vacuole that they take in. And what's interesting, the phagosome then merges with a lysosome. And lysosomes, I don't know if we talked about those too much, but those were organelles in eukaryotic cells that have specific chemicals, or they have basically a solution in it that is able to kill a microorganism. So it helps with digestion, but also eliminate microorganisms, etc. And so they destruct and eliminate those with those phagolysosomes. So it's a combination of a phagosome, a vacuole that the thing comes in and it fuses with the lysosome. And I show you.
The lysosome is the cell, or the lysosome?
No, that is a cell. It's a organelle. So if you look at it here, you would have this is the macrophage. So you have a few other organelles. What's that one called? Huh?
Golgi apparatus?
No, the ER, the Golgi. Well, maybe you saw this one, this down here, connected to the nucleus. Anyway, so you have those lysosomes that help with all kinds of other things too, like digestion. But there's there's basically chemicals involved that would be toxic, but they're produced in those lysosomes. And then if you have a cell, so you have a cell and there's like, say, this viral or this, I think in this case it's just some microorganism, and it has, as you can see, these, as you cannot see, these these pathogen-associated patterns on it. And so what happens is the receptor of the macrophage can actually recognize it, and then they get internalized, and then basically this internal phagosome is fused with this, toxin-containing lysosome, and then it kills the microorganism in these vacuoles, and then the little particles that are not harmful anymore are kicked out. That makes sense, right? I mean, that's the way they do it. I didn't come up with this. So I can do it one more time if you want. Really?
One more time.
Okay, so chemotaxis doesn't matter right now. Just imagine you have a microorganism, which in this case would be, have these microorganisms, bacterial cells with these protein structures on their membrane that are part of a pattern. So this is not a specific, this big macrophage is not specifically looking for one structure, one specific structure. It's just looking at some patterns. It says, okay, I recognize this one because I have these cellular receptors, the toll-like receptors, for example, or the pattern recognition receptors. So just imagine the macrophage is a white blood cell that has the ability to recognize a microorganism. So it recognizes it, and then it takes it in. So it binds it here, it takes it in, and produces this phagosome. Because it's a phagocyte, it has a phagosome, right? And then it also has lysosomes, which are vacuoles, so enclosures that have toxic to microbe toxic substances in it. I think peroxide is probably part of it, or peroxide could be part of it, as far as I remember. And so when they combine those together, a phagosome and a lysosome, you have a, what's it called? Phagolysosome?
Yes.
Perfect. And then the phagolysosome does what it does best. It kills the microbe and it throws out the particles. So these steps are just, this is done, this is the combination, and then death, out. Yeah? Okay. So we'll go through two more things. I don't know if do you still have some kind of order in immunology in your head? First barrier, second, and then I just don't want it to confuse with all these other things.
We're still in second.
Yeah, we're still in second. And so this was just an example of those macrophages, for example, not specifically binding, what you just saw here, not specifically binding one very distinct structure. So if you go to the third line of defense, those white blood cells have the ability to, for example, just recognize a protein. They don't need a whole bacterial cell. They don't need all these other cell structures or membrane structures of microbes. They need sometimes just a miniature piece, like one protein, and they can attack it. So that's where it's specific, and that's what needs to be learned by the immune system, whereas this is still the innate, the general immune system.
So the PAMs were saying that it saw something like a cell wall, right? And then knew that it was foreign, right?
Yeah, what did you call it? The
PAMs, the PAMs.
Yeah, those are the pathogen-associated patterns. So it just means
So PAMs is something specific that like a lot of different bacteria will have, right?
Yeah.
Okay.
That's
like
the difference. Yeah.
Okay.
So that means your immune system probably before it got to a higher step, it was basically able to say, usually I can recognize a microbe. I know what they look like. I know they have certain, say, proteins, glycoproteins or lipoproteins on their membrane. They have certain structures that we can recognize. And then through evolution, it wasn't enough, so somehow the immune system evolved to getting more specific, and also creating these memory cells, so you have an immune reaction immediately after being exposed to a pathogen twice. So it's a progression, I would say, and this would be more in the beginning, or earlier in the evolution of the immune system, where you say there's a general pattern, what these microorganisms display, and we know how to react to it. But then more specifically, later on, there are now the B and the T cells that can actually recognize. Number one, they can recognize much more fine structures, and then they actually, and I said that before, one cell there's only a fit 1-to-1, so only one cell can recognize one specific structure, and that cell then gets a lot of clones out, and they get rid of that pathogen. But we'll get to that after the next break. So interferon and those are the complement and the interferon, and I said those are just know that I want you to have heard of it, but that's basically a way of signaling to neighboring cells what's going on. So you have interferon alpha, beta, gamma, different ones used by different cells. They're small proteins, and so basically if you have a cell that is infected with a virus, it would synthesize some interferon and then send those out, another way of signaling it. And then if a neighboring cell gets those interferon signals, it would then immediately produce antiviral proteins, or it would just be prepared to synthesis, and it would be prepared to work or to fight against the virus rather than being attacked and not being prepared. So it's a very local system. So interferons are not, they're just signaling molecules or proteins that cells that are neighboring other cells are using. And I don't think I'm going to say much more about that, because the immune system is complicated enough. You don't have to know too much. If you hear interferon therapy or something, sometimes people you can through biotech you can start producing those and maybe injecting those, having people or having it's part of a therapy you can use interferons for, but in comparison to what the other white blood cells do, it's not as important to know about those. And then the complement is a little bit more important because it interacts with the specific immune system. But here you basically just need to know, so complement are proteins in the blood, and they're basically helping. So they're in the blood and they recognize, again, like other white blood cells, these are not cells, these are just proteins, but they recognize or they can bind to microorganisms and then make the immune system more aware of there's a microorganism. So there's three different pathways how they go. So I don't want you to worry about remembering those, but if your blood comes in contact with a microorganism, they can go one of these three ways, and basically you would, for example, have a mannan, which is a sugar. So you have a sugar in the cell wall of a fungi or other microorganisms, and this complement would recognize it or basically bind to that sugar. So you have a protein that just binds to that fungus cell.
Why?
because it recognizes it. It's so complement is just a bunch of proteins that are also able to bind. So instead of the difference between the interferon and the complement to all the other cells is that these are just proteins that help. So complement actually means complementing the immune system. So it's another system to help the immune system recognize microbes. So instead of a cell wandering around trying to figure out is this a microbe or not, these proteins, we just produce them, I guess, in our blood, are able to recognize. They can bind to a sugar. So it's just a protein that binds to another structure. So it binds to a sugar. It binds to other receptors maybe, or it binds to a normal cell wall of a microorganism because we don't have cell walls. So it's able to bind to a cell wall of a microorganism, and then by doing that, it can, number one, destroy that organism on its own, or two, it can make it. It can basically flag it and say, here is a microorganism. So it's just a system to help the immune system. And the only thing I really need you to know is that there's a complement system. There are proteins. They can attach to specific structures. Microbial structures or eukaryotes, and what they basically do is if they don't just alert, they usually alert the immune system, but they also are able to build these through a process, a 4-step process. They're able to, through a cascade, build these porins. So they basically, in the membrane of microorganisms, they're able to build this hole into, or to make these holes into microorganisms and then destroy them that way because they eventually die. So let me see if I'll get back to that because I know it's a little bit much, but the complement system it's important later on. So during a viral infection, which of the following defenses is most likely to be active? I have to think myself, Oh, didn't we have a viral in there? Viral infection. Yeah, I would probably be C. As far as what we've done so far, the virus that, because inflammation is more, is coming from a injury, and then you have fever, which is part of it's just the most likely to be active. It would probably be the interferon. If you have a virus infection, it creates the synthesis of interferon. I want to go back to the last slide then. Are we going the right way? Just the last one and this one. So we've covered these physical, chemical, and general genetic barriers in the first line of defense, PS. And those were the second line were the inflammatory interferons, phagocytosis, and complement. And then after a 5-minute break, we can go into the third line and look at B and T lymphocytes. Does that sound good? So even that's important to know is there's going to be some major differences between those, and you should have heard of all of this, but it's not. I'm not going to ask, or you don't have to remember all of the very small details. Once you go back and read over it, it's probably making a little bit more sense. The first time, it's a lot of different cells and proteins.